In this video, we will continue our discussion about chemoresistance in pancreatic tumor cells. So in the previous video, we have understand that the HA in the high help matrices cause the chemoresistance in the PDAC. So moving further, the researchers from the literatures have found out that HA-CD44 interactions has also been reported in several papers to cause chemoresistance in other cancer cells. So they decided to check this HA-CD4 interactions in these PDAC tumor cells, whether it also causes the chemoresistance in the PDAC tumor cells or not. So what they did is they first checked the CD44 expressions in the high help matrices and the low help matrices. That is shown in the panel A, where we can see that in the high help matrices they see that more CD44 RNA expressions compared to the low help matrices. After that, they also checked the protein CD44 protein expressions using flow cytometry, and they found the similar results that in the case of high help help matrices, they see that more CD44 expressions compared to the low help matrices. And these things they have validated using the immunostaining, where the CD44 is been labeled as a pink dye, where we can see that in the low help matrices the CD44 expression levels are lower compared to the case of high help matrices, where CD44 expression levels are more. After validating that in the high help matrices there are more CD44 expressions, they wanted to check whether this CD44 HA interactions cause this chemoresistance in the PDAC. So the approach that they took is to knock down CD44 in the organoid, pancreatic organoid, and then culture them in the high help and the low help matrices to check the chemoresistance. First, they generate this PDAC tumors using PDAC tumor cells with this CD44 knockout using CRISPR Cas9 system, where they use Cas9 with CD44 guide RNAs to knock down CD44, and then they use those PDAC organoid to culture in the help low matrices and the help high matrices, and thereafter they did the drug treatment to check the marker expressions. And what they have found out is that in the panel E they have validated this knockdown of the CD44, where they can see that in the wild type the CD44 expression levels are high, whereas in the knockout CD44 expression levels are low or negligible. And then they did this culture this PDAC organoid, in which the CD44 is knocked down in the high help matrices and the low help matrices. And what they have found out is that organoid diameters is similar in both the cases, that is high help and low help matrices. That is shown in the panel F. And when they check this CD44, and when they check these CD44 expressions, what they have found out is the CD44 expressions are lower in the case of CD44 knockdown, as it is expected since CD44 is knocked down, compared to the wild type case, where in the wild type case we can see that CD44 expression levels are high in the high help and are low in the low help matrices. And since previously they have also concluded that this ABC2 drug efflux transporter gene is also responsible for this chemoresistance in the tumor cells. So they wanted to check whether this CD44 expressions also regulates these ABC2 genes or not. And what they have found out is that in the case of the CD44 knockdown, the ABC2 genes are down regulated, and there is negligible, or there is no statistical significant difference between wild type and the CD44 knockdown cells in the low health matrices, and whereas for in the case of high health matrices, they can see that there is a statistical significant decrease in the CD44 knockdown case compared to the wild type case for these high health matrices, indicating that CD44 also regulates this ABC2 genes interactions. And thereafter they wanted to check this matrices about this they wanted to check this CD44 knockdown PDAC organoid with the drug treatment, and drug that they use is gemcitabine. And what they find out is that in the case of wild type, they can see the statistical significant difference for the single cells, and they did their drug treatment experiments on the single cells. So for the passage 4 in the wild type, they can see the statistical significance difference in the cell viability between high health matrices and the low health matrices. In the case of high health matrices, they see that more gemcitabine is required, and these things have been quantified in the panel I, where we can see that for the wild type, the gemcitabine IC50 requirements are statistically significantly higher compared to the case of these low health matrices. But when they did the same experiment on the CD44 knockdown single cells, what they have find out is that there is no statistical significant difference between the high health matrices and low health matrices in the case of this normalized cells, in terms of this normalized cells viability. And these things are quantified in the panel I, where they can see that in the case of CD44 knockout, these gemcitabines IC50 requirements is for both the cases almost similar, or we can say there is no statistical difference, indicating that this CD44 HA interactions is the initial step towards the chemoresistance in the PDAC, and this CD44 HA-CD44 interactions might somehow also regulate this ABC2 ABCG2 genes that act as a drug efflux transporter. So in this experimental results, they have successfully defended their hypothesis that HA-CD44 interactions cause chemoresistance in the PDAC tumor cells. If you need to understand more about their experimental details about this figure and these experiments, or some other experiments in this paper, or if you want to check the supplemental figures, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.